So here we are dealing with the MSc2nd Sem botany. The chapter is Sphagnum, and we are looking into the life cycle. The gametophytic stage. The external features of Sphagnum. The juvenile stage is protonema and is formed by the germination of spores. It is fixed with the rhizoids. Rhizoids possess oblique septi. Stem. It is erect, covered with false leaves and short branches. Very short internodes on the stem are featured. The stem has two branches, divergent and drooping, or flagelliform branches. The stem is featured by the cortex, hadrome and medulla. Cortex. It is the region of compact tissues, multilayer 2 to 5. These cortical cells bear a spiral thickening and the oval perforations and store water by capillary action. Sphagnum molluscum and tanellum show the cortical cells. They are elongated to form the retort cells. These are the retort cells. Retort cells, which are distinguishable, and it is the specific feature here. They are not found in Mertensia and Pelia, Anthoceros, etc. in other bryophytes. So they are different from Mertentials, Anthoceroidals and Jungermannials. And the present-chimatous region, that is similar to that of the parent-chimatous region. It is a cylinder of narrow, thick-walled, elongated cells with 4 to 6 layered region. It provides mechanical support. Mechanical support starts from this kind of TS structure which you see here inside the stem. Mechanical support is concerned here. As you see the hard xylem and secondary tissues in the advanced flowering plants, angiosperms. The somewhat harder kind of cellular organization is seen is the advanced feature, which is supporting the stem, the aerial part, medulla. It is composed of thin-walled, colorless, parenchymatous cells. It looks like pith of the angiospermic plants. Pith and functions as the storage region. Leaf. It is one-celled, thick, rectangular, and they are equal size. The leaf possesses hyaline and asymmetry cells, you see here. Hyaline and asymmetry cells which are green and photosynthetic. The asymmetry means photosynthetic. So it is somewhat similar to those of Markin shells. The leaves are highly different from the other bryophytic forms. As you see the structure here. Reproduction. Vegetative reproduction takes place by innovations which arise and as a special vegetative branches grow upwardly and they develop into a new plant. Second, the multiplication of protonemal branches are developed by the marginal cells into the green cellular filaments or the secondary protonema from the leafy gametophyte. Third, regeneration. It is a regeneration of the damaged tissues, whether they are fragmented and damaged or the damaged tissues on the gametophyte. As a result of desiccation, it suspends respiration and photosynthesis, and thereafter the gametophytic plant is developed. Sexual reproduction. It takes place by means of antheridia and archegonia. As usual, the sexual reproduction takes place by the developed antheridia and archegonia. Antheridium, these are found as catkin inflorescence, in the form of the catkin inflorescence. So catkin-like branches as the male branches are here with foliage leaves. The antheridia are stalked and globular shaped. They are somewhat longer. You see here. It bears spirally coiled biflagellate antherozoids. Antherozoids are developed by the endocytes and they are liberated on maturity when the antheridium absorbs water and swells up. Second, the archegonium. These are 1 to 5 developed at the tip of the short archegonial branches. Pericuteal leaves protect archegonia. It is somewhat similar pattern on the side of the archegonia of Markensia. They are covered with the pericutium inside the arc structure in case of Markensia. An archegonium is developed by the archegonial initial cell, and this initial cell later forms the central cell, which ultimately forms the venter and the neck. The matured venter has an egg, and the neck has the neck canal cells and the cover cells on the top. It is longer with 8 to 9 neck canal cells. Fertilization, it is usual, takes place in water. Water is essential as that of the other bryophytes. The cover cell opens. The cover cell, these are 4, they open and a passage is made. In the neck is opened here chemotactically. The egg is fused with the antherozoid chemotactically. Later, 2X zygote is formed on the sporophyte. The sporophyte, it is differentiated into foot and capsule. The seta is compressed. Pseudopodium is found. The capsule is unique and different from other forms. The capsule possesses convex and curved arc-like spore sac. The spore sac is arced. The spore sac is only one, and that is covered by the jacket wall. Annulus, operculum and calyptra are found. Columella is over-arched. By the spore sac. The epidermis contains chloroplast to photosynthesize. It is the outer ring, outer covering, protective ring. Non-functional rudimentary stomata are found. Stomatas are not found, but rudimentary false stomata are found here to assimilate air, gases. You see the life cycle pattern. Alternation of generation is here. The gametophyte, two gametophytes will be here to give rise to the archegonia and antheridia. You see, fertilization takes place later on, and the zygote is formed, the dyad, tetrad, and later on this massive structure is formed, multicellular embryo is formed, and that develops into the sporogonium. The sporophyte is here. The sporophyte with calyptra, operculum, spore sac, columella, foot and vaginella. Pseudopodium is here, but it is somewhat different from the polytrachum and funaria. Polytrachum and funaria are different. So therefore the marsupials, juggernails and anthoceratops and other forms of Brady are different here from this kind of sporophoric structure, and it's a dehiscence as well. The mechanism of dehiscence is also different. When the spores are germinated under the favorable conditions, the spores absorb water and swell up. The extend ruptures and the intine forms a germ tube that forms the green filament as the protonemal branches. The protonemal buds give rise to the gametophyte, and the alternation of generation takes place where the sporophyte is dependent on the gametophyte. The sporophytes or the sporophyte of Sphagnum and the sporophyte of polytrachum have major differences structurally and functionally. The mechanism of dehiscence is too different from the standpoint of creating pressure and dispersal of the spores. So the spores have absorbed water and they have swollen up, moved towards the archegonial neck. Ultimately the fusion takes place Later on, the zygote is formed. The zygote has developed into the sporophyte, and the sporophyte has developed the descent mechanism. The descent mechanism in the form of the cloud of the spores. They are dispersed along with the emergence of cloud. So it is somewhat different mechanism. The sporogonium, particularly the capsule, bears the clouds forcibly. The role of columella is here, that juts the spore sac and the upper column to liberate the spores in the form of the cloud. And after all, the spores are developed in the protonema and protonemal buds, and they develop the gametophytes. That's all. Thank you.